you are made of, you got skin, bone, you get tendon, some hair, you got a lot of stuff there. That's all made of what? Proteins. That's proteins. Where do proteins come from? And what are proteins? Just some basic stuff. Does anybody know what proteins are made of?
Amino acids.
How many amino acids are there?
26.
20. So, proteins basically are beads on a string. Beads on a string, strings of amino acids, like beads on a string. The final protein is folded in a three-dimensional form, which actually determines its form. But you can think of them as beads on a string. So where are they made? Where do they come from? They're made in cells. Cells make proteins. And I'm going to give you a basic metaphor for the mechanism of a cell. Because everything that you do, all of your activity in your body happens in a cell. Everything. Your nerve, your thinking, your muscle activity, it happens in a cell. The metaphor is, think of a cell as being a medieval city. A medieval city has a wall around it with gates into the city. It has a castle, and the castle has a moat around it. So that's our metaphor. The membrane has gates, which in a cell are called receptors, and they have keepers at the gates that guard what gets in and what gets out. The nucleus, the castle in the cell is the nucleus, and like I say, has a moat or a membrane. Take a handout, please. Take a handout. There are manufacturing plants in this city, and in the cell they're called ribosomes. They're scattered throughout the cytoplasm, and that's where the proteins are actually made in the manufacturing plants. There's recycling centers. In the cell they're called lysosomes. They recycle all the compounds that are used in the cell. There's energy plants. This is the basic structure of both a plant and an animal cell. We know that the plant cells have chloroplasts that animal cells don't have. And the chloroplasts start the energy cycle by making sugar from the sun. And the mitochondria are the energy plants in both plants and animals that take that sugar and turn it into a compound that is then fuels all the chemical activities of the cell. So those are the energy plants. The food storage, we have grocery stores and food storage units, and in the cell they're called vacuoles. You have an inner membrane structure in the cell. This is a big round three-dimensional thing. They are the roads and the bridges, the infrastructure, the transport mechanism. Take a handout. Take a handout. The receptors are the post office. They control the interchange of messages. And I got down here tax collectors, but I haven't figured out what to sign up to. I'm sure there's got to be tax collectors. So it's a dynamic, busy. Think of it as a busy, teeming city, because it is. It's teeming all the time. When you're sleeping, your cells aren't sleeping. They're teeming. Your cells are restoring what you used up during the day. Restoring the energy. It's teeming. And I missed one. The enzymes. The army of enzymes. The people. The jobs are enzymes. And enzymes are also proteins. So the proteins of your hand are structural proteins. But the jobs in the city, all the activities of the city are regulated, facilitated, run by enzymes, which are the action proteins. They're also proteins. So everything that you have going on in your body is mediated or made of proteins. So it's a busy, busy place. And all these units are interconnected. It's an interwoven ecosystem. It's a little ecosystem within a bigger ecosystem. which is what we're talking about today. So, what goes on in the castle? DNA, think of it as a twisted ladder. Two strands of linked compounds that are just the same all the way along. It's extremely long. If you had stretched it out, believe it or not, every DNA in every one of your cells would stretch out to about 2 meters. Unimaginable, but that's what they say. The only distinguishing features of this twisted ladder, strings of these compounds repetitive, are on the steps of the ladder. At the ends of the steps of the ladders, of each step, is a compound called a base. And there's only, in DNA, there's only 4 different bases, and they go by the first letter of their name. So there's A and T, C and G. A is always across from T in the ladder, and C is always across from G in the ladder. And they can be on either side of the ladder. And that's the only thing that distinguishes the difference in the genes, what we call genes. 4 bases. Now, DNA is what codes for proteins. So how do we get from these 4 bases, these 4 letters, to these proteins, multiple different kinds of proteins? That is what's called, it was figured out in 1953 by Watson and Crick, almost a half a century ago, basically half a century ago. They figured out what they named the central dogma. I didn't give it that name, but I'm glad they gave it that name because it really is dogma. But to them, it was the whole picture that DNA codes, makes RNA, which codes for proteins via the fact in all of life that 3 of these bases in sequence, in a particular sequence, codes for one particular amino acid. In this example I have here, I didn't tell you that when the DNA is transcribed to the first copy of RNA, which is just transcribing, taking one strand and then transcribing the bases over to one of the strands. It just changes T to U is the only difference there, basically. RNA is a little different strand, but the T changes to U, so, and you have still 4 bases in RNA. UGG, in that order, codes for tryptophan, one of the amino acids. So now we know all the code, the 3-letter code for all the 20 amino acids. Well, we know what life is all about, right? That gives us our sense of understanding of saying, well, now we can manipulate life. We know the nuts and bolts. It really fits in well to the mechanistic model, the worldview that life is described by the idea that life is a machine composed of nuts and bolts. That's where our scientific community is steeped in. Analytical science means you analyze down to the smallest parts. And once you have the smallest parts, you know how it ticks. And boy, does this fit into that. And it sure gave us our culture, our society, which is steeped in analytical science, the mechanistic viewpoint, it sure gave it emphasis that now we know the code, we can play around with it, we can study it, we can learn a lot from it, and we can manipulate it. I'm going to come back to this later. There's a lot more to say about that. So now we're getting into what you came here for probably, is how genetic engineering is done. First, let's just look real quick at plant breeding, which is limited by nature. In the flower of the plant, the pollen from the male part has to arrive at the female part, and then it grows a pollen tube down into the ovary, where it then fertilizes the seed in sexual reproduction. There's all kinds of natural barriers and limitations to keeping foreign pollen out of this reproduction system. In fact, our taxonomical system of identifying plants, humans have established these taxonomic classifications, family, kingdom, species, is based on form. So if two plants have a different form, they're in different categories, in different species. If they have the same form, then we know they share genomes. If they have a different form, then obviously they don't share genomes. So that tells us right there which plants cross-pollinate, the plants that we put in separate categories, species. So the limitations of nature that we've described by our taxonomic system tell us that the plants don't normally cross-pollinate outside of species. They cross-pollinate within species. There's a little bit within genus. There's always a few exceptions, but that's the general rule. Genetic engineering is not subject to those limitations. It happens in the laboratory, and it uses a method called tissue culture that scientists have learned how to take individual cells, these dots represent individual cells of a plant, in a petri dish. And they can give it the nutrients and stimulants that they can cause a cell to grow into a complete plant. Say, a potato cell taken from the leaf of a potato, say, they can get it to grow into a complete plant. So that was the starting point. So let's say we're going to make a genetically engineered potato to kill insects. So you need a trait to put into these cells, single cells. So the trait to kill the insects is taken from Bacillus thuringiensis, and the Bacillus thuringiensis has a toxin, natural toxin, that kills certain species of butterfly and beetle. So they are able in the laboratory to isolate the portion of the DNA of that bacteria that expresses the toxin in the cell, using the mechanism of the cell. And so they can isolate that and multiply it and put it in a beaker. So it's swimming around in a beaker. So, theoretically, you could pour that beaker into the petri dish and maybe get it into the cells. But it won't. You can't just do that because it won't go into the cells by itself. That DNA that you've isolated from bacteria. You need something to take it into cells. What in nature has the capacity to invade cells? What else? Disease organisms, pathological organisms have that capacity to get in through the post office, to get in through the septors, to get in across the moat, and cause the mechanism of the cell to replicate itself. And that's what they use. The vector, the carrier is taken from pathological organisms. The most common is in plant genetic engineering. It's also pathological organisms in animal and human engineering, other organisms. But in plant is Agrobacterium tumefaciens, which is a bacteria that causes tumors in plants. So they isolate the part of the DNA that in that bacteria that gives it the capacity to get in the gate, and multiply it and put it in your beaker. And so then now you've got a trait and vector, and actually DNA tends to hook up with other DNA because the ends of it it's kind of like Velcro. One end is a hook and the other end's a loop. So it'll hook up. And so now we got a beaker of these two. So theoretically, we could squirt this in here and the vector would carry it into the cells. But you also need something called a promoter. Here in this picture, when you're in the nucleus, when messages come in to say, we need a protein. The messages go to an enzyme protein, and that does the transcribing from DNA to RNA. It goes chunk, 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 chunk. It hooks to the DNA and then chunk, 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 chunks along. Other enzymes bring the materials it needs, and it manufactures this as it chunks along, moves along the DNA, using energy from the mitochondria. So the promoter is a stretch of DNA, just a stretch of, 8, 10, I don't know how many bases that this enzyme recognizes as the starting point. And it chunks along, and then it stops at the terminator. So you need that section, that stuff that tells the cell when to make, to promote, to express this package. And again, where do we get our promoter for plant genetic engineering? It's taken from a pathological organism. CAMV is cauliflower mosaic virus. And the cauliflower mosaic virus is almost universally used because it is promiscuous. It is a very promiscuous promoter. It'll promote almost any package in almost any plant. And it promotes at way beyond the regulatory capacity of the host cell. It promotes strongly to make sure that this gets expressed in the host cell. And that can be very disruptive. It's also known as take a handout. Are you Shepard?
Yes, I am.
Hi. You made it. That's Shepard Ogden. Welcome. So it's also known as a hotspot for jumping around and landing in other places. In fact, it landed in the corn in Mexico. That's what they found in the corn in Mexico, the cauliflower mosaic promoter. For sure. It was the rest of the studies that are being criticized, but they didn't criticize that the cauliflower mosaic virus was found in the native corn varieties. So you got your promoter and terminator, and then you need one more thing. So say if we squirted this in there and it got inserted into the cells, it's really haphazard. I mean, a lot of it won't get inserted in the cells. A lot of cells won't have the insertion happen. You need some way to determine which successfully work. Take a handout. So you need a marker, what's called a marker. And what they use is antibiotic-resistant genes, also taken from bacteria, so that this is a complete package. You squirt it in there, the vector takes it into the cell, the promoter promotes it, and when it's successfully inserted and working, then later you squirt this petri dish with a antibiotic, this antibiotic, and if the whole package is working and expressing, take a handout, it won't die from the antibiotic. Is that clear? Did everybody get that? So that's basically it. That's how they do it. And then only about maybe 1% of these cells will be successfully expressing the package. And then they grow those cells out to plants. Say we're making a BT potato. Maybe 1% of those that we're growing out actually are good potatoes, because there's a whole bunch of them going to be freaks, going to be messed up. Because there's no control over where in the genome, host genome, this package lands, obviously. There's no control over how many copies land in the host genome. You can have massive scrambling of this. You can have scrambling of this construct as it's going in, especially with the cauliflower mosaic virus. You can disrupt the host genome, depending on where it lands. And plant defenses. Plants have enormous, complex, amazing defenses in all aspects, and also in the nucleus. And plant defenses for foreign DNA can be activated, and that's called gene silencing. And that can be activated down the road after
this plant has been grown out and maybe reproduced, there's still the gene silencing can happen and does happen. And then post-translation processing, well, when we come back to this, maybe we'll talk about that. So those of us who are concerned about this trend in our society say it is inherently unpredictable and dangerous. We don't know what we're doing via this, what I've described basically as genome disruption, but also via what is known as horizontal gene transfer. The microbial world, bacteria and viruses, exchange genetic material naturally, be outside of sexual reproduction. They don't exchange it. They can exchange it for reproduction, but they exchange it what's called horizontal gene transfer, just coming up beside each other, and one says, ooh, you got something interesting, give me some of that. And so they trade it around and then go off, and without making any babies. So that's the problem in medicine. That's why we have our diseases coming back resistant to our antibiotics. It's because, more and more, it's because the bacteria share genes. So what are we doing here? We're putting all these pathological organisms out there in the biosphere in mass. This is in every cell of every plant out there in those fields. Antibiotic-resistant genes out there. We're encouraging the spread of antibiotic-resistant genes by this method, and the British Medical Society has come out with, they've stated they've calling for a moratorium on the release of GMOs just because of that issue. And many other organizations have also called for that. So that's basically how it's done. Now I want to come back to the central dogma. The basic assumption is that it's strictly linear. It was a half a century ago. Take a handout. The handouts are right there. And it's still the assumption today that DNA makes RNA, makes a protein, and nothing else matters. It's the nuts and bolts, and we can shift around the nuts and bolts, and there's no other effect. Well, since 50 years ago, we've learned a heck of a lot. Even before Watson and Crick, in 1953, who coined the central dogma, we knew that there are mobile elements that just move around here, which kind of makes a little chink in the armor of the central dogma. And then later we learned about retroviruses. The viruses, unlike bacteria, viruses are just a bit of DNA with a protein coat. And retroviruses are a bit of RNA with a protein coat that actually make a copy, get into a cell, make a copy backwards of DNA, and then insert themselves into the mechanism of the cell, into the DNA. That's a retrovirus. HIV is a retrovirus. So that refutes the central dogma. And then things we're learning is, in your cells, your DNA, 1% of your DNA actually codes for proteins. 99% of your DNA is other stuff. A lot of that other stuff is something called introns. I have a little loop here that actually is a loop outside of the twisted ladder. And we really don't know a whole lot what all this other stuff is. The geneticists, the corporate geneticists, call it junk DNA. Well, I don't think anything in nature is junk myself, and I'm certain that 99% of that has some function, but it's ignored. The other thing that refutes the central dogma is humans. The Human Genome Project just completed in February of 2001, we finally found out that we have somewhere around 30,000 genes. But we know functioning in our bodies are somewhere around 300,000 proteins outside of the immune system, which has umpteen million proteins, capable of producing umpteen million proteins as antigens. Just forget that. We have about 300,000 proteins in our bodies, but they, as the Human Genome Project says, only 30,000 genes, actual things that code for proteins. So that also refutes the central dogma. So what is going on here? Well, all of these colored circles are enzymes that have a job, a job in the city. To get from here to here, you have a lot of things going on. This enzyme transcribes. This enzyme cuts and pastes. It cuts out the other stuff and pastes together the final RNA product called messenger RNA. And this one is a spliceosome. It also can splice in different ways. And then we have a transport enzyme, and this one, then it's translated or transported out of the nucleus to the manufacturing plants at this stage. And then you have enzymes that come along and translate the code from 3 bases to 1 amino acid. And a lot can happen in that stage. And then you have another enzyme that cuts and pastes that cleans it up. It actually knows how to take away the mistakes when mistakes happen. And then you have the enzymes that fold this string of beads into its final form. Like I said, it's a 3-dimensional form, and any change in that 3-dimensional form changes its function completely. It's essential to have the exact right 3-dimensional form fold. And added to that are some elements, copper, sulfur, vitamins, whatever, that are essential to that 3-dimensional form. For instance, the prion that is in mad cow disease, they consider it a misfolded protein that's causing all this problem. So all these steps in the process of getting from here to here, from DNA to a protein, happen in the whole mechanism of the cell. And it's a complex pattern of multiple feedback loops. How do these enzymes know what to do? We don't know. It's a huge mystery. Multiple feedback loops, which include signals from inside the cell, from outside the cell in the larger organism, and from the environment itself. The system is a mystery. We don't know what we're doing, and you can only call it intelligent. I mean, it's so intelligent. Enzymes even know how to fix DNA. I call it intelligent and awesome. It's a whole system. So, and our approach of our society, coming at it in the materialistic, mechanistic worldview, refusing to look at everything that refutes the mechanistic worldview, is what we have to change. It's a whole system. It's an ecosystem within ecosystems, which we need to learn to understand. And the mechanistic view really fits into the corporate agenda, that everything is separate, and that you can patent, and that you can change the nuts and bolts. And there's where we have to really, really work. So given that the central dogma is a fallacious assumption, what is keeping this going? Well, it's a gold rush. We're back to the 49ers gold rush. Money, venture capital. In the year 2000, in the United States already, there were 1,280 biotech companies that had sprung up. Some of them, before they even got their office open, had tens of thousands of dollars of venture capital coming at them. In that year, they raised $35 billion in venture capital. Anybody who hung up a sign and said, "I'm a biotech company," had it coming in. But even below that, what keeps this gold rush going? It's based upon two policies that are followed by our government. One is that now we have the patenting of life, and the other one is called substantial equivalence. The patenting of life, we never patented living organisms in the past. Before 1980, it was policy and standard tradition that living organisms were not a novel invention by human beings, were not patentable. Patent means something you've invented, an innovation, and living organisms were creation of nature. But in 1980, a bacteria was kind of put together from 3 other bacteria, and the company, GE of all things, because we call this GE, asked the patent office, U.S. Patent Office for a patent. Well, plants have had the PVP protection, plant variety protection for seeds, for seed varieties, which was through the USDA and didn't cost a huge amount, and allowed the farmers to save their own seeds and allowed the breeders to share the germplasm of the plants. But patenting does not allow those two things, and it gives the company a lot more control over the finances associated. This application for a patent of a bacteria by GE finally went to the Supreme Court in 1980, and the Supreme Court changed the question. They changed the question about it. They said the issue is not, is this an animate versus an inanimate thing, but can an animate thing be considered a human creation? And they said yes, in a 5-4 vote. And that cracked the door open. And the ag chemical companies saw that crack and stuck their foot in and started buying up the pharmaceutical companies, the seed companies, and started this massive trend in biotechnology to genetically engineer food plants and have them be distributed. In 1985, the Patent Office submitted to this pressure and patented the first sexually reproducing plant, a corn, and that continued to this day. And we'll hear more about that in the forum tomorrow. Shepard will give us a good breakdown of the history of patenting in the first thing in the morning. Saturday, thank you. And then the other thing that keeps this gold rush going is the policy of substantial equivalence. That's our regulatory agencies. In 1989, the National Research Council made the statement that genetically engineering produces something that is basically no different than traditional breeding, and therefore doesn't require special laws, special regulations, or special testing. And then that was adopted by the FDA in 1992. So we really don't have any safety testing of these inherently dangerous products. I'm going to stop there and see if you have any questions, see if I've completely flummoxed you or what. What do you have?
I've read that about 65 or 70% of the world's soybean crop now is genetically modified. So, do you know anything like what kind of modification that is, how it can spread so fast?
Okay.
What chance there is of having not genetically modified soybeans?
The 4 crops that are genetically engineered out there in mass are soy, corn, cotton, and canola. The 2 kinds of engineering are either herbicide resistant, and most of that is Roundup resistant, but there are other herbicides, or BT, which we talked about a bit. The soy is Roundup Ready. It's all Monsanto's Roundup Ready soy. And yeah, you're right, 60% is, around 60% of what's grown is Roundup Ready. About 30% of the corn, about 60% of the canola or more, but canola is completely lost. There is no more non-GMO canola, I don't think. At least that's what Percy Smizer says. And a lot of the cotton, 60-70% of the cotton. Soy is a little different from corn and canola in that it's in the bean family and it pollinates itself. So the spread, the drift of pollen, like what happened with corn, Starlink corn and the corn in Mexico, is not as big a problem. But still, segregation has happened. And in fact, I heard from a farmer in North Dakota who spoke at a conference I was at who lost a $10,000 market of soy, and his was non-GMO soy, because somehow it got contaminated with soy. So it is a big problem, and if you want non-GMO or organic soy, you really have to research your source.
And how can you tell? Let's say you had some soy. How would you find out?
There's a relatively inexpensive test that the grain elevators do, more than the large processors do, to see if there is any telltale. Basically, what they do is they have an assay that looks for any of the remnants of any of those 4 packages that are part of the process, and they will do that on a sample of a truckload or a sample of an elevator load or a sample, and it's gotten to be fairly regularly done now because of the problem, as Jill mentioned, of segregation. Since soy is self-pollinated, the problem isn't so much that it's spreading in nature field to field, but that the companies that are dealing in it are selling it to more and more of the farmers, and the elevators that store the soy between the time that it's harvested and the time that it's distributed for food use or feed use haven't been sufficiently careful about keeping the non-GMO and the GMO supplies apart. And probably what happened to this farmer is that the truck that it was transported to the elevator in wasn't sufficiently cleaned out and might have had a couple of beans from another crop, and one of those fell into the sample. Or it could be that even the crushing machine that was used to crush the sample to get it, and I don't know how they actually do the sample, whether they crush it from the slurry or what, but it could be contamination at any step of the process. Probably his soy itself was not contaminated when it left the farm, but by the time it had worked its way through what the dealers call a commodity process, it had somehow gotten admixed with a very small percentage of somebody else's crop that was GE, and that got into the sample. The buyer based their decision on the test that was done on the sample and rejected his load, even though it may not actually have been contaminated itself. But the contamination that's happening in the soy market is happening in the marketing, transportation, and storage, not in the growing, where the spread of the acreage, the increase in the acreage that you referred to, is happening because of market forces of selling the seed, not by crossing in nature.
Are there any countries that have
that's the big thing about wheat. We expect wheat to be coming out shortly, and the major importers of wheat have said they're not going to take any. They're not going to take any. And wheat is our big export. Half the wheat grown in the country is exported for human consumption. And that's the big concern of the farmers is they're going to lose their market in the United States if GMO wheat is released in the next couple of years. It hasn't been released yet, but that's one of the purposes of our forum on Saturday, is try to figure out how to stop the release of GMO wheat. Yeah, a lot of countries, Europe has tried to, the whole European Union has tried to stop all of it. They haven't completely stopped it, but they've put up the most determined fight. And I think 19 countries now require labeling. Not in the United States. Yeah.
Well, they haven't approved it yet. Last I knew, Japan wasn't accepting GMO soybeans.
Yep.
say whether there's been some real analysis done on the proteins of any of these strains?
Yeah.
How different the protein is?
Yeah. For instance, very little real safety testing is done, but some has been done. You may have heard of Arpad Pushtai in England when he was doing his research on potatoes. He came out and said, well, there's some major difference in the actual content of the potatoes. He said that there was a significant difference in the protein between GMO and non-GMO potatoes, the protein, starch, sugars, and enzyme content of his potatoes. And then you have Monsanto's Roundup Ready soy, which has been tested. It has changes in fat, carbs, ash, fatty acids, choline, which is a vitamin, was reduced 29%. Trypsin inhibitor, which is a potential hay fever, allergen, was up 27%. Lectin was up, change in phytoestrogen content, and it had 2 extra genes.
My question has to do not with the mix of those things, but the protein structure itself.
Oh, you mean the final product?
Yes.
I don't know of that. Does anybody know that the final product, say if you put a gene in, say if you put the Bt toxin in, do you get a different toxin or something?
Yes.
Is that your question?
Yeah.
You get an enzyme that, you get a new enzyme that makes that toxin, so there's a new protein in it.
Here's an argument that I used at a hearing on GMO wheat in the last legislature.
Introduce yourself.
I'm Ron Erickson. I'm a House Representative here in Montana. And I was simply a co-sponsor of the bill. But I went down to give testimony right after I heard testimony to go ahead and give great tax breaks for alcohol plants using wheat. And the way an alcohol plant works best is that you go ahead and use the protein, the extra protein for animal feed. And so the argument that I made was that because the protein was going to be different from this GMO wheat, that we were going to be feeding it to cattle, and that we didn't know what sort of results might occur when you had very, very new protein and fed in a concentrated way to cattle. I thought the argument sounded okay, but I wasn't sure, so I wanted to hear from somebody about whether or not that's another argument to make, because what it does is it brings the cattle growers into the same concerns from the wheat growers.
Good point. Bill, would you like to take that one? This is Bill Tracy, University of Wisconsin.
Yeah, there are new proteins there that have never been there before. And of course, in the example you're giving, those proteins would be highly concentrated. Now, usually and not always well enough, these, not in the concentration you're talking about, but in just the regular corn or the regular soybeans, these have been fed to animals, and allergenicity tests have been done. But it's very hard to do an allergenicity test. What they do is they see how quickly the protein breaks down. And that's what happened with the Starlink corn, is that was a different Bt protein, and it broke down more slowly than the other BTs. They already decided the other BTs were okay. But the Starlink, they decided was not okay because of the rate that it broke down in this assay they did. So there are different proteins, there are new proteins that have never been there before, and whether they've actually been evaluated under the conditions that you're talking about would be a case-by-case basis, and I'm sure in some cases they wouldn't have been.
Thank you.
I think one of the other concerns with feeding some of these proteins animal feed is the antibiotic resistance markers that go with it. There have been some, I don't know if it's been done outside of laboratories yet, but there have been some demonstration and transfer of antibiotic resistance through gut bacteria and with these mechanisms. And that, you're feeding antibiotics to cattle all the time in feedlots, and now you're feeding them antibiotic resistance genes, and there's a whole thing going on there that we really don't know the consequences of, I think.
I just want to say that the general argument about these protein issues is that they're just proteins, and proteins are broken down quickly in the diet. That's what people usually say. But the point of allergens is that not all proteins are broken down quickly. So whether this new protein is broken down quickly or not has to be evaluated by someone.
And it hasn't been, is what you're saying. I mean, that's the...
Well, the thing with the BT protein, again, is the reason Starlink was not allowed as a human food is somebody had evaluated that and realized that it was broken down. The length of time taken to break down the Starlink protein was long enough to put up a warning flag. So somebody did do that test.
But if we're particularly concerned about the GMO wheat part,
Right.
then it hasn't been that. Okay.
Let me challenge you a little bit here. Ron Newland, Western Plant Breeders, with PhDs work with me, and they're unanimous on the other side of this argument. And I'm trying to keep an open mind here and try to figure out the right position. You, Bill, with the corn project. Recently, there's been research showing that, well, we know that vomitoxins in corn natural carcinogen, to the point that most corn is used not in food consumption because it doesn't get the food FDA label stamp its credit. So BT corn comes out. BT is a natural organic pesticide. Correct.
In its form as a pesticide is actually the spore of the bacteria, and that is different from expressing the actual toxin in the plant. It's different.
Well, what I'm getting at is that the majority of allergens and viruses, etc., are natural.
Everything's natural.
Right. Good point. Agree with you on that. So now with the advent of BT corn, I'm challenging us. With the advent of BT corn, there are now studies coming out that show the vomitoxin levels are being reduced within corn.
I'll just explain what's happening is there's a lot of these fungi that make actually very nasty toxins are introduced by insects. And the insect coming in and eating the corn introduces wounds that these fungi grow in. And what Ron's saying is that BT prevents the insects from making the wounds, so in some cases those fungi are reduced.
So in total, we might be healthier.
In the case of, those are very nasty toxins, the fusarium, vomitoxins.
Yeah. And see, where I'm coming from here is we got to be careful here because we're dealing with life, we're dealing with illnesses, we're dealing with bacteria, everything else. God bless you for bringing these issues up because they need to be understood. But what are the alternatives?
Well, you bring up a good point. The problem is we are steeped in something called industrial agriculture. And we're long down the road of industrial agriculture. And to me, industrial agriculture is oxymorons. You cannot apply industrial
values, which is basically yield and efficiency. Yield and efficiency. Getting the most for the least amount of money, the least amount of human effort, to agriculture, which is living organisms, ecosystems within ecosystems. When you start doing that, you start breaking the patterns of nature. So we are so steeped in industrial agriculture that we don't even know what we're doing. We don't know how to get out of it. This whole process is just continuing industrial agriculture. It's continuing the dependence on chemicals, because what do you got here? You got herbicide. Roundup Ready is so you can spray Roundup, so the companies can sell more Roundup. That's dependence on chemicals, and BT is just making the plant express a pesticide. We've forgotten how to do ecological agriculture. This whole thing just continues that whole process. We've got to break that whole pattern completely. It's also contributing to the consolidation of agribusiness. We now have just a few big agribusiness, we call them the lords of life, who have bought up the pharmaceutical, the ag chemicals, the pharmaceutical, and the seed industries. And it's all consolidating into just a few. And that's happening in a lot of the sectors in our society, in medicine and a lot of other sectors. That's what we got to break and change. And we've had this green revolution, what we called the green revolution, for 50 years. Well, 2 billion people are abjectly poor in the world, and that is growing. This green revolution has not fed. The green revolution has fed industrial agriculture. And the industrial agriculture has fed these lords of life, these giant corporations. That's the problem. And this is the technological fix. This is saying technology and corporations can solve our problems. I disagree.
I'm hoping to speak directly to one of your points. Jerry, I'm an organic farmer by trade and also in the seed business. And it's true that BT is an accepted organic control. And it's true that BT has the proper paperwork with the EPA. But as every farmer knows, and as every registered pesticide applicator knows, every single pesticide is registered for particular uses on particular crops in particular ways. So the fact that BT is accepted as an organic control in no way affects whether or not its incorporation into the genome of the plant is safe or not. Because if that was true, then the simple fact that malthion has a lower LD 50/50 than rotenone would mean that we could spray it on everything. But the EPA doesn't allow that. It's a crop-specific and an application-specific kind of regulation. Now, the difference is
that one too.
Well, of course there is, because I don't agree with the EPA on all of these things. But I'm only trying to point out a slight mushing of the argument that went on there. Wait,
I think you ought to be proud of what you've done here, that the pesticide poundage in the Midwest is way down because of BT.
Well, I think BT is a very effective control, but that's my point that I want to make next about that. Because the reason that BT has been an effective control, especially for organic growers, is because of the application method that's allowed. That application method, by definition, is incomplete coverage because it's applied externally and it's applied on a case-by-case basis and it's applied repeatedly but not to the entire environment of the pest's action. When you incorporate the BT into the genome of the target plant, you essentially affect 100% exposure of the pest when it attacks the plant to the control. And what you do is you decrease the evolutionary response time of the organism that is the problem so that resistance develops. We've been able to use this material for a very long time without the development of resistance. And the industrial metaphor under which it's being incorporated in the plant, this is Jill's argument, but I'm trying to phrase it to you a different way that perhaps in the seed industry we would understand it. Once you do it by using this industrial metaphor, what was an effective and long-term sustainable control gets into a situation where it is no longer sustainable over the long term, except as it becomes part of a product cycle, which is very valuable to people in my business. And perhaps your PhD friends, I have a number of them myself, because what it means is that just as is done with most chemical controls, by varying the regimen of controls that are applied, sort of as a mimic of what we do in sustainable ag and crop rotation, by rotating the chemicals that are applied, and by constantly developing new variations on the 100 or so BT toxins that exist within the genome now, you're able to have a product cycle that will indeed give us a certain period of time of control at great profit to the owners of those things, but at eventual long-term destruction of the efficacy of the product in its non-commodified form. And that's the problem. That's what Jill's really objecting to, is the taking of a natural process and, through the industrial metaphor, commodifying it so it becomes an input that the producer is completely dependent on and can only get in a cash transaction, which is great for those of us in business.
Yeah, this is interesting because you're coming from a profitable standpoint.
I've been in business for 30 years doing this.
I know we're after food safety and protection of the environment first.
Well,
so
one
is
of
she,
the things
but-
you said was misinformation.
You're thinking aflatoxins were dangerous than...
There's
You
plenty
said
of
that there has
misinformation
been
going on
a
here.
big decrease in the poundage of chemicals in the Corn Belt. Well, that's because the chemicals that were used were these low-dose chemicals. IMI is a low-dose chemical. They spray it by the ounce on corn. And Roundup is a poundage chemical. You know, they spray it by the pound. So they
with Roundup Ready, or I'm sorry, they replace it with BT where they don't have to spray, but IMI is a low dose, and but the Roundup is making up for it because you're spraying more chemicals with Roundup. Yeah, and they're having to spray it two and three times. It is true. Chuck Benbrook did the study, and it's in one of those websites that I pointed out to you. The solution we're promoting in this workshop, and that most of the organizations involved in the globalization issue are promoting, is local food systems. Build local food systems. Get off the corporate food grid. Our food travels 1,300 miles. Basically, what we buy at the grocery store has traveled 1,300 miles. Yeah, there you go. You know, we can build local food systems, grow it locally, consume it locally, grow it organically. It can feed the world. That's how it fed the world up until now. I mean, the people that we're running off their land because of our industrial agriculture have been able to feed themselves in the past better than they are now.
Where the pests come from and how they relate together, and that's what we're talking about, is understanding what's going on. And when you walk out, I've been in a cantaloupe field in Guatemala, for example, that's been sprayed in a 60-day crop treated with a systemic chemical. It's actually absorbed by the plant that kills whiteflies for a few weeks, and then sprayed once a week or 3 times a week for the duration. That crop has had like 20 sprays of chemicals on it by the time you're over, trying to kill whiteflies. You walk into that field and there's 2 things alive in that field. One is the cantaloupe plant, and that's pretty marginal sometimes, and 2 is the whiteflies. There's nothing else alive out there. And so they call me up and say, help, how do I kill my whiteflies? Well, what else have you done? And in that case, you got to think about space and time. They plant cantaloupe in a continuous cycle for 8 months in 5,000 hectares, which is roughly 12,000 acres of land in one valley in Guatemala. And at this certain time of the year, probably a third of the cantaloupe in the country are coming from 3 areas in Central America that do this. And it's all, in Guatemala, it's owned by 9 farms, 9 companies, Del Monte and Dole being the 2 biggest. And so you got one place, whiteflies love cantaloupe. I mean, they just think cantaloupe is one of their choice foods. And these are an introduced whitefly that came in on something else from the Middle East. And what happens is you plant that host for 8 months in one monoculture in one place, and you start out with one whitefly, and pretty soon you got so many, literally 500 per square centimeter on the bottom of it. You turn a leaf over and it's white, and the plant dies shortly thereafter. And we made the suggestion, they want to know, okay, chemicals aren't working, we'll bring in the biologists. So I go off to Guatemala and spend weeks stomping around in the fields. And the solution is readily apparent. I mean, there's nothing you can do to stop that level of whitefly infestation by treating that system. You can't do it. I mean, they did it with the worst chemicals you can think of, and you kill 91 female whitefly laying eggs on a 2-leaf cantaloupe plant will be responsible for 8 million offspring by the time that cantaloupe's harvested. Okay? And so you start out with 10, you're done for. I mean, you got to have 99.99% control, and it isn't going to happen. And so the solution here that we suggested, we got all 9 of these guys together in a big room and had a workshop, and the suggestion was, okay guys, this isn't working. Us biologists can't save the system unless you don't plant cantaloupe for about 30 days, because the life cycle of a whitefly is 20 days. Okay, so you just take away their host for 30 days, and that will probably solve most of your problems. And you know, I mean, it's not all of it. There's host plants in the woods and stuff like that. I mean, you have to think about the whole system. But the problem is they couldn't do it. The investors don't let them. The packing house doesn't let them. The import regulations, the pesticide, they just could not. You couldn't get 9 farmers in Guatemala who were losing money. I mean, the farm that brought us down there at the height was packing 110,000 boxes of cantaloupe a week. And when they brought us down there, they'd packed 3,000 boxes that week. Okay, I mean, they were heading for out of business. In 2 or 3 years, they will no longer grow cantaloupe in this valley unless they change their thinking in space and time. So this is not just crop-based. You got to think about the whole system. And that's basically, there's 3 things in a take-home message here about what is the buzzword floating around now is knowledge-based agriculture, ecological agriculture, or agroecology. There are different people who are calling it different things, but fundamentally it comes down to 3 points. And this system, you know, when you think about it and when you see examples of it, it can work to feed the world. This system is not. You know, I mean, that's a given. With we're going to have to try something different, and this is what people are talking about. An agriculture that, number one, is locally adapted. You can't plant the same variety of cantaloupe or the same variety of wheat or same variety of anything everywhere in the world, because climate's different, bugs are different, diseases are different, soil's different, dirt's different. There's a guy that I heard speak not too long ago in California that runs a seed bank in Iowa or Kansas or something that collects variety, all kinds of vegetables around the world and tries to maintain diversity. He has in his seed banks 3,000 genetically distinct separate varieties of tomatoes. That's where you solve the problem of whiteflies in Costa Rica or caterpillars in Florida. You don't solve that by planting the same variety of Roma tomatoes all the way from Florida to the other end of South America, which is what we do. And so that diversity's out there, and that's why people are working so hard to keep it, because that's where the local adaptation comes from. There's hundreds and hundreds of varieties of potatoes in Peru. There's people who do nothing but try to keep that diversity alive. And what do we eat in this country? 3 varieties of potatoes, white ones, red ones, and russets, right? Well, there's, what, 300 or 400 varieties of potatoes, many of which are resistant to Colorado potato beetles, which we spray the hell out of in the United States every year. The second thing which follows from that is the system has to be diverse. You can't plant monocultures. You got to be diverse in crops, you got to be diverse in the system, and you got to be diverse, as I was saying, in time and space. You got to think about the whole system. And finally, you got to be sort of holistic, and this is coming back to dirt. You got to understand everything that's going into the system, the biology of, and the relationships. And I don't know how many farms I've been on where somebody has sprayed for bug A and bug which kills the predators of bug B, and bug B comes and eats your crop. And that's happened in GMO cotton all over the place. I've worked in a lot of genetically modified cotton. With the BT cotton, it really does wonders for killing caterpillars, because caterpillars are killed by the BT gene. But it turns out that they used to be spraying for the caterpillars, and that was also killing the lygus bugs. Well, lygus bugs aren't susceptible to the BT genes in cotton, so now you spray for lygus bugs instead of caterpillars. And there's a lot of debate about it, but fundamentally the amount of chemicals on average in cotton hasn't changed much with the introduction of BT cotton. You just spray for different bugs. And so you got to look again, you're not looking at the whole system. And I don't know how, there's textbooks written about this, and I mean, Miguel Altieri wrote probably one of the first serious textbooks about this, and you can read them online or buy them or whatever, but you got to understand the system where you live. You can't import, Montana's not ever going to be very successful growing, pineapples for obvious reasons of weather, but for the same reason, a lot of temperate climate crops really don't fit very well in tropical climates. And that's what we do in the name of development. A lot of cases is we go and, all the cantaloupe in Guatemala was originally a USAID funded economic development project where you take a temperate climate crop like cantaloupe and make it grow in Guatemala so you can send it back to the US. And for a real taste test one of these days, go to Albertsons and buy a cantaloupe from that's been shipped 3,000 miles from Central America, and go get one of the melons at the market from Dixon and taste them. And one tastes like a cantaloupe and one tastes like cardboard, I guess. I'm not sure. That's probably enough of me talking. We should continue some more discussion or questions, or is this making sense? Or I apologize for being a little disjointed with this, perhaps, because we were going to try to share this with Miguel, but that didn't happen. So I may be giving knowledge-based agriculture a bit short shrift here, but.
How do we get from here to there?
That's one of the big themes that we're going to discuss for the next few days. And I see a number of, well, the first thing we got to do is take control of the research agenda. I mean, Jill talked about this a lot. I mean, the amount of money being spent by both government labs and private corporations on GMO crops is astounding. The amount of money spent on understanding how different agricultural models in different parts of the world actually function is tiny. And we got to get some control over where we spend our resources. And that's one very specific discussion on Saturday is what the corporations are doing buying off the land-grant universities in this country. I mean, that's, Monsanto runs around and makes a contract with Montana State and North Dakota State and I don't know who all to breed GMO wheat. And when they do that, the public funding goes away. So that's part of a simple-minded answer, I guess. But it's comes back. We've got to take control of the institutions that govern these decisions, and that's what these summits and protests and whatever else people are doing to try to get some attention to these issues are fundamentally doing is demanding to participate in some way in the allocation of the world's resources to problems. And I looked it up one time. I'm not sure I can remember the exact numbers. When Monsanto and Aventis and a couple companies were doing genetically modified corn, they went around and just bought up all the what they call a foundation seed company, Holden Foundation Seeds and people like this. These were small family-owned companies that basically provided the commercial genetics to Pioneer and the people who made hybrid corn seed. And if you're going to do GMO corn, the first thing you got to do to make money is make sure that nobody else can do it. So they went out and bought all of, 3 companies now control the commercial corn genetics for 80 million acres of our biggest crop in the United States. And I know somebody who used to work for Holden Foundation Seed. This is a company that had about 120 employees and maintained that genetic stock. Monsanto just upped the ante until they sold, until it was $1 billion that Monsanto paid for that seed company. I mean, I know a guy, they just gave $1 million to every one of their employees and everybody shut down. And the point is that $1 billion used to corner the market is more than was ever spent by anybody ever on the basic fundamental biology of insect pathogens, understanding what in nature actually kills insects. Okay, and that's what I do for a living, is find out pathogens that kill bugs. And nobody has ever in the rest of the time spent $1 billion trying to understand those systems. And we need to do that. I mean, the resources allow us some people to corner the market, and other research just doesn't get done. And that's what needs to happen in these knowledge-based agriculture systems. There's examples out there of really wonderful systems that function beautifully that we need to understand more about. The most beautiful farm I've ever been on in my life was in Nicaragua, up in the mountains. And I know some really fine people who are tropical agricultural research folks and that worked on that. And they went to an area, deliberately picked an area to develop this farm that had been virtually sterilized in the production of cotton in Nicaragua in the 70s. And they quit growing cotton in Nicaragua because they could no longer control whiteflies, one of my favorite bugs. And they had sprayed so many pesticides on there and then walked that this was basically a wasteland. So these people had set out to restore that with a local understanding. And what they did is it was a big farm. It was a cooperative. They had about, what's a manzana? Oh, Lord, it's about 1,000 acres that they had as part of this cooperative. And they grew 6 or 7 crops. but they did it in strips. They thought about it. And then, you know, they were trying to learn and improve all the time. They grew tomatoes and corn and beans, onions, sesame, cotton, and a couple other crops, I don't remember it all, and had thought about their system. And they planted those in strips, and some of them for export and some for local consumption. They were trying to, you know, feed themselves and make some money. So they divided it up between, you know, corn and beans is for the local market, and sesame and onions, or they export onions to McDonald's, for Lord's sake. And I can't describe the whole system, but there's one thing I noticed. You walk in there and it's alive. There's bugs, there's beetles, there's birds. I mean, it's buzzing. And you walk into that cantaloupe field that's been sprayed, you know, 67 times, and it's just sterile. It looks sterile, it sounds sterile, it smells sterile, and it is sterile. But you walk. They thought about it. I mean, whiteflies are not a pest of corn, and they are a pest of tomatoes and peppers and a few other things. Okay. So what they did is they always planted the corn upwind from the tomatoes. That's all it took, because whiteflies get blown. They're little bugs, and they blow on the prevailing wind, and that's how they're designed. So you plant the corn upwind from the tomatoes, all the whiteflies get trapped in the corn, and that's what it took to control whiteflies. Sesame actually produces some allelopathic compounds, some compounds that suppress other plants for a period of time. So they would rotate the sesame through for weed control. So they had this system, and onions repel some other pests. I mean, they really worked this out, and you got crops, strips, that they had a rotation thought through in time and space and rotation, and they were growing cotton up there without insecticides. And people were critical. It took them a few years to get their yields up because they started some really lousy soil. But when they finally got done, their yields were every bit as good as the neighbors down the way that were using the chemical systems. Their input costs were probably a half, and they were making money. It was the other thing, beside the most beautiful farm I've ever been on, it was the only farm in 3 provinces in Nicaragua that was actually making a profit. And it was very labor intensive, but so was all agriculture there. And that model may not fit Montana, or it may not fit Argentina, but it fit where they were. And that's the key. It depended on what they were growing, 6 to 12 rows, depending on, you know, what. And they, you know, they had to make some compromises around some of the tilling equipment and things they used, but generally 6 to 12 rows of strips. And in a lot of cases, strip crops might not make any sense. You know, if you own a big bud tractor and live in northern Montana and try to farm 40,000 acres in one piece, it's a bit tough on the equipment allocation. But it works in that system, and that's, you know, they understood that piece of land. They could tell me which way the wind was blowing, which way the rain came from, what way the sun came from, which trees they needed over here, which plants outside the boundaries over here harbored which beneficial insect that controlled which bug in their crop. I mean, they understood what was going on in that 1,000 acres. They knew it like the back of their hand. And that's the difference. That's where we need to spend our resources, is understanding how to make those kind of systems work, just understanding how they function. Sorry, got to quit talking. You got a question?
Well, I was just going to make a point. You know, I think research is important, and I think the kind of ideas you're talking about are interesting and could be used in all sorts of circumstances. But the real question was, how do we get from the left side to the right side? And the problem that I really see is in making that transition is number 3 on the left side. Is the farmers are in a vice, and it's very hard for them to move. And I think the issue, beyond the research, the other issue is how can we create systems that allow farmers to do. If we gave them a good system, how can we get them to do it? How can they afford to do that?
It's actually in the process. If you did like a 3-piece box of the food system, the first box is the input sector, the chemicals, machinery, etc. The middle is the farm, and the third is the downstream processing, wholesaling, retailing. I'm sorry, I forgot you. Brilliant presentation. As you pointed out, the piece, 5 cents of wheat in a box of Wheaties, 3 cents of corn. The farm sector is being scrunched, and the other 2 sectors are growing. So what the you know, whatever you want to call it, knowledge-based agriculture movement is doing is saying, okay, how do we reduce that input sector? We go organic, we reduce our machinery costs, we bring back horses, you know, get people on the land to do physical labor, et cetera, et cetera. And then we do direct to the consumer, community-supported agriculture, where you have 2, 3, 4 dozen families that contract the farmer directly. In San Francisco, we have a thing called the box. And it's a subscription service. And they bring a box once a week onto your front steps. It's all organic, it's whatever in season. So you've got to take whatever's in season and learn how to cook accordingly. But they're making a living. They're making a good profit on good land and doing restorative agriculture and showing that it can work, it can be profitable.
One of the things I've been trying to understand, that I certainly nowhere near understanding it yet, is how far up can you apply those kind of principles? And particularly thinking about GMO wheat in Montana. I mean, we're being presented with an alternative that farmers are going out of business. If you use GMO wheat, you'll save, you know, $1 an acre on your herbicide cost or whatever. And you may. The economics of Roundup Ready wheat are not at all clear. But even assuming you saved a little bit of money on that, that just means that you're participating in sort of that race to the bottom in the price of wheat, because somebody in Argentina is going to find some other way to cut their production cost just a little more, buy some more land, and pretty soon the price of wheat that, you know, Continental or ADM or somebody will pay you $2.90. And it seems that we need to think about community-supported agriculture models on a global scale. I don't know if you can think about it in terms of wheat, but that's one of the things I'd like to try this weekend is, can you? That rather than fighting about GMO wheat, Monsanto's answer, I mean, again, you know, I've pointed out at any number of meetings that GMO crops and saying, well, you know, your customers don't want to buy this. And in the world of genetically modified crops, this is the only industry I know of anywhere on the planet where the companies that do it are happy to put their customers out of business. I mean, they sue them, they run the prices down, they blow their export markets. I mean, you know, it doesn't make any sense to me that you would try to sell something to your customer that's going to put them out of business. But what we need to, you know, maybe rather than participate in that continued race to the bottom, we ought to think about what Montana's wheat customers really want. Japan buys hard red spring wheat from Montana because that's one of the few places in the country that really grows good hard red spring wheat that makes the kind of bread the Japanese want to make with it. Okay, so maybe rather than participating through, you know, letting ADM or Cargill sell that wheat to the Japanese, maybe Montana ought to say, we're GMO free, we're going to keep it out of the state, and we're going to figure out how to downstream capture that value for that wheat in Montana. Okay, we got big sky organic best bred wheat in the world, and maybe the Japanese would pay $3.50 a bushel directly to Montana for it instead of going through the, you know, we got to think about those kind of systems on some various different kind of scales. It exists to a certain extent in some fair trade coffee, but it still needs to be a much larger thing, I think. But you know, we need to figure out how to do these things.
How large a difference the international ban from Europe, European Union, and various places in Asia, they have banned genetically modified foods. How much of a dent does it actually put into these big corporations' budget?
Well, that was Monsanto's answer when I've ever raised this question in meetings, is their answer to that problem is they're going to go lobby and try to control the governments in Japan that make those decisions, you know, rather than it. But most European countries have some restrictions, or if there's not outright restrictions on the importation of GMO crops as government regulation, there is, in a lot of cases, effective banning because they have labeling laws. So consumers sort of decide that, and a lot of grocery store chains in England and Europe and other places, you know, advertise we don't have GMO crops, et cetera. So that market force actually keeps a lot of it out, even though it may be legal to import certain kinds of GMO crops in some instances. The regulation of it internationally is very complicated. And we're getting into issues now like what is GMO-free soy, and different countries are establishing, you know, we'll tolerate 3% contamination because you can't get GMO-free soybeans anymore. And so we're getting in all kinds of regulatory nightmares this way that we need to avoid with wheat if this poor state is going to survive. I mean, half the wheat in the United States is exported. Montana, it's about 90%. And almost all of that goes to countries that will not import wheat because of the kind of GMO crops. Because of the kind of wheat we grow, it goes to Japan and the Middle East and some to Europe and places that do not like GMO crops. This, I mean, wheat is by far the biggest cash crop in the state. I mean, the next one down is pretty small. Cattle are the biggest thing we do, but in agriculture. But wheat, I think in the year 2000, Montana exported $175 million worth of wheat. And you know, that's a big number in a small economy. And if we, you know, the price of wheat was $2 instead of $3 a bushel because we lost our markets for it, that would hurt.
Organic isn't in there. Because you don't have to be totally organic.
Yeah, I mean, it's sort of, if you do real knowledge-based agriculture, you're probably going to end up as organic. I mean, I do a lot of work with organic growers, but I tend not to think of in the U.S., I mean, we've got a particular definition of, a legal definition now of organic, which is a definite improvement over not having some definition of organic. But just being organic does not describe knowledge-based agriculture. And I've worked in, in California in the last year or two. There's a company that probably packs, T&A probably packs a quarter to a half of the head lettuce in the United States. And they have organic lettuce. I mean, they grow organic lettuce, but they grow 1,000 acres or 2,000 acres of organic lettuce and call in the biologist to say, oh my God, we got bug X. I mean, they've done it as organic, but they're not doing it, they're following the rules, but they're not understanding the system. And so they get in almost as much trouble as the chemical guys that way. The one thing that saves them is they tend to get a higher price for their lettuce or whatever. So I tend to not try to think about the letter of organic and trying to think about systems agriculture. Usually it'll come out okay if you don't. And there's some real issues with U.S. organic certifications and things in exporting countries, in Latin America in particular, having some big debates about, you know, because some of the ways U.S. organic rules don't really fit the way you grow certain crops in Brazil or somewhere. And so there's some big debates about that. But so I tend to just organic will follow if you think about the system, I guess, is the short answer.
Is it my misperception, but like the entertainment industry, the gap between what the producers get for something and what the consumer ends up paying for keeps widening, and there's this whole group of middle people in between. I see this in the lumber industry too, where there are these wholesalers, and if you figure out between what people get at the start of this and now if there's value added to all this stuff, okay, I can see it, but if they're the type of materials where there isn't value being added, I don't know how you cut out these middle people. I don't know how they got into being. Well, originally they were legitimate. It's like it's gotten out of hand.
Well, I mean, yeah, there's certainly, if you're going to trade foods, you got to pay somebody to move tomatoes or wheat from point A to point B. But a lot of those disparities have been the result of deliberate policies in countries like the U.S. I mean, we have been preaching free trade. That's a fairly recent innovation in American thinking. We've had severely protected agricultural markets in this country and still do. I mean, one of the biggest hassles my Nicaraguan friends do is we have embargoes on Nicaraguan peanuts, sugar, beef, and sesame. Because
you all vote for these certain individuals and make these...
We tend to, yeah.
So what's going on?
And so, but you know, there's a really interesting book I've been reading lately. There's a Latin American writer named Eduardo Galeano, and he's an Uruguayan guy, brilliant writer, it's unbelievable. But he's written a book, it's a little bit dated because he wrote it in the 70s. It's called, what's the Open Vein, thank you, The Open Veins of Latin America. And the subtitle is 500 Years of the Exploitation of a Continent. And he describes in there the policies over the years that importing countries use to keep value of imported crops. I mean, and some of these still exist. I mean, we would not allow, Brazil tried to make, one of his examples is when instant coffee became the rage, Brazil produces most of the world's cheap coffee that gets turned into powdered instant Nescafe. And through tariffs, economic pressure from international institutions, loan financing, and all sorts of other ways of extorting one government, extorting another, we prevented Brazil from making powdered coffee. So that Brazil exports coffee beans to the U.S. and we make Nescafe or Nestle or somebody makes powdered coffee. There's nothing real magical about making instant coffee. I mean, it's not a real technologically sophisticated process. And by government, the ability of rich countries in the north to impose their economic will on poor countries of the south, we did that to coffee. We've done it to cacao, chocolate. I mean, Africa was for 100 years not allowed to make chocolate. There's no reason why you can't make chocolate in Africa, but it's all made in Belgium, and that's because of the way the tariff rules were put in place. And so you can't just stop and say, okay, time for free trade now after you've done that for a few hundred years. And you know, most countries in the north that got rich by protecting their markets. And then they preach free trade, but it's, and those things still go on. I mean, we make it very difficult. I've worked in tomatoes in Morocco, and the Europeans have all kinds of ways of, the Spanish in particular, keeping Moroccan tomatoes out of Europe except for a few months of the year where they can't grow them in Spain. And I was part of an AID project that was supposed to be helping Moroccan tomato farmers grow more tomatoes. Well, the problem wasn't growing more tomatoes. The Moroccan tomato farmers grow tomatoes better than anybody else I've ever seen in the world. The problem was they had no market for it. So AID was going to spend all this money and invite us to conferences on improving tomato production in Morocco, and all they needed was, well, if you want to export tomatoes, was to lift the protection in Spain. I mean, there was no technological issue. For me to teach a Moroccan tomato farmer how to grow tomatoes was pretty ludicrous. I mean, those guys had it figured out pretty well. I mean,
Chris, many of the problems that you expounded upon eventually come back to the question of who has the power to make these things happen, either directly or indirectly through control of the media, and so as to prevent true knowledge from being applied for the good of the people.
well,
And so politics in one sense or another is involved in this.
and it's even in a lot of cases, this is why these institutes, the IMF, the World Bank, and particularly the G8, who act sort of as a board of directors for all this, are so important, is because a lot of this functions by, I don't know what the right word here is, it's extortion. I mean, this is an example I've lived through in Nicaragua. I mean, I mentioned coffee. I mean, Nicaraguan coffee farmers are going out of business in droves, and it's mostly small farmers, and when they go out of business, they either cut down the trees or they come to Managua, and Managua's overcrowded and poverty-stricken, and you know, it's just the classic thing. And Nicaragua's government at the time was a pretty serious mess, but at least the legislature in Nicaragua passed a law that put a 2-year moratorium on bank foreclosures on farms, coffee farms less than a certain size. I think it was like 5 or 7 acres or something. It was pretty small. And the legislature passed that bill, and some friends of ours that work in the Nicaraguan Support Network followed this process through the system. And the Inter-American Development Bank, which is one of the big economic offshoots of the World Bank, went to the president of Nicaragua and said, look, if you sign that bill, which is anti-competitive and anti-free market and anti-whatever, we will withhold your next $50 million loan package. Okay, and Nicaragua needs that next $50 million loan package in order to apply for its debt relief program, because they're up in international debt up to their eyeballs and can't pay their debt. So they, so that's how that's done. I mean, that's not some magical thing. It's the IMF can go to some place and say, you do it our way, or we withhold your money, or we apply this pressure, or whatever. And also now there's people literally starving to death in Managua because they'd have, if you're making $1 a day seasonally picking coffee and you lost that job, you're in pretty bad shape. And Nicaragua exports food to the U.S. and it's there's no reason for Nicaragua not to be able. There's no reason for anybody to be hungry in Nicaragua as a result of the lack of food.
Thank you.